Hello students, very good evening to all of you. I'm your biology educator Dr. Meetu Bhavnani. Welcome to Vedantu NEET English channel. Today we are going to deal with the unit Plant Physiology of class 11th, one short series for NEET 2026. Chapter name is Photosynthesis in Higher Plants. This chapter this chapter is from lower classes. We are continuously discussing this chapter Photosynthesis in Higher Plants since very small classes, like we have studied in third, fourth, fifth and continuous, but now in uh, from NEET point of view we are discussing in a very broader way. Let's show in my mobile. Hello. Hi everyone. Hello everyone. Now, what do you mean by photosynthesis in higher plant? First of all, what do you mean by higher plant? Higher plant means developed plant. The parts, the morphological parts are very much developed. Root is very much developed. Stem is very much developed. And leaf is very much developed. Now, photosynthesis occurs in the leaf part of the plant. Okay. Let me check the voice. So, photosynthesis occurs in the leaf part of the plant. We very well know that photosynthesis, green plants carry out photosynthesis, a physico-chemical process by which they use light energy to drive the synthesis of organic compound. Here, inorganic compounds converted into organic compounds. Okay. Now, ultimately, all living forms on earth depend on sunlight. For every energy, for every driven energy, you require sunlight. For every driven energy, you require sunlight. Okay. Now, we require sunlight. Uh, Producers require sunlight for synthesis of food, organic food, and this producers going to become food for herbivores, and herbivores become food for carnivores. So means sunlight is very important. Photosynthesis is important due to two reasons. It is the primary source of all food on earth. On earth, it is the producers who are manufacturing food, who are manufacturing food. Okay, who are manufacturing food. So the food is now going to, exchange with the other organism in the form of energy. It is also responsible for the release of oxygen into the atmosphere by the green plants, because here CO2 plus H2O is using, and oxygen plus organic food has been formed. Okay. Now what do we know? We have already know that to carry out photosynthesis, inorganic materials are present, sunlight is present, but very important, green color pigment, chlorophyll should be present. This chlorophyll is actually trapping the sunlight. Trapping the sunlight. Okay. A variegated leaf or a leaf that was partially covered with black paper. Now, if you are taking this leaf, okay, and if you are taking another leaf, this is the bare leaf which is continuously getting the sunlight. Cover the leaf with a paper. This part will perform, uncovered part will perform photosynthesis. Covered part will not perform photosynthesis. Why? Because chlorophyll is present in complete leaf, but The covered part in that chlorophyll is not trapping the sunlight. Okay. Hi. Okay. Now that means for the green means for carry out the photosynthesis, a mandatory things should be happen in the substrate side. Inorganic material CO2, H2O, then sunlight, chlorophyll. Okay, these things should be present. Then only oxygen will released and organic food will manufacture. Now, so many experiments are there which actually shows the the which actually shows the importance of substrate, importance of substrate, complete procedure of photosynthesis. Okay, now this is done. Now, early experiment Joseph Priestley performs a series of experiments revealing essential role of air in the growth of green plants. Role of air, role of air in green plants. Okay, so a bell jar. Now here a bell jar is there, a leaf is there, lighted candle is there, burning candle is there. Okay, now he concluded that a burning candle or an animal that breathes the air, but both somehow damage the air. But when he placed a mint plant, then the mouse stayed alive and the candle continued to burn because here light is coming, oxygen is producing. Oxygen is producing. Priestley hypothesized as follows: plant restore to the air whatever breathing animals burning candle remove. Okay. Now, whatever the oxygen they are, releasing, utilized by that particular green plant, and in turn oxygen is producing, which are survival for the mouse. Remember, he would need to rekindle the candle to test whether it burns after a few days. Now, using a similar step as one used by but by placing it once in the dark and once in the sunlight, Jan Ingenhouse showed that sunlight is essential to the plant process. Okay, sunlight is essential to the plant process. Purify the air fooled by burning candles. Okay, later he identified these bubbles to be oxygen. Means in the procedure of photosynthesis, on substrate side CO2 gas is released and on the right side oxygen gas is released. But in the case of breathing and exchange of gases, opposite is there. Oxygen is inhaled and CO2 is exhaled. It was not until about 1854 that Julius von Sachs provided evidence for production of glucose when plants grow. Glucose is usually stored as starch. Glucose is forming and storing in the form of starch. His later studies showed that green substance in plants is located in special bodies, that is chloroplast. He found that green parts in plant is where glucose is made and the glucose is stored as starch. So starch is a polymer and glucose is a monomer. So many monomers form the polymer. Okay, done. Now consider the interesting experiments done by T.W. Engelmann. T.W. Engelmann. Okay. Now, he illuminated a green alga cladophora in the suspension of aerobic bacteria. He observed that bacteria accumulated mainly in the region of blue and red light of split spectrum. The first action spectrum of photosynthesis was thus described. Okay. By the middle of 19th century, the key features of plant photosynthesis were known. Now, plants could use light energy to make carbohydrate from CO2 and water. I have already told you. Okay, now what will be the empirical formula? Let's see. See this, CO2 plus H2O light, CH2 plus O2. Now, these are represented a carbohydrate. This is representing a carbohydrate. Empirical formula of a carbohydrate. Done. Glucose, a 6-carbon sugar. A milestone contribution to the understanding of photosynthesis. Okay. Cornelius Van Niel, who based on the study of purple and blue-green bacteria, demonstrated that photosynthesis is essentially a light-dependent reaction. Essentially means the presence of light is mandatory in the case of photosynthesis. Okay, done. This H2O light. Now in green plants, H2O is the hydrogen donor and it oxidizes to oxygen. Means the oxygen source is H2O in the photosynthetic reaction. Some organisms do not release oxygen during photosynthesis. Some organisms do not release. And H2S instead is the hydrogen donor for purple and green sulfur bacteria. Okay. Hence, he referred that the oxygen evolved by the green plant comes from H2O, not from carbon dioxide. Question comes in NEET 2026. Source of oxygen in photosynthesis is H2O. It is not CO2. Okay. Easy question. So 6H2O plus 12H2O light, 6H2O 6H2 plus oxygen, hydrogen donor. Hydrogen is donating, so oxygen is patching. C6H12O6 represent glucose, storage of glucose starch. The oxygen released is from water. The oxygen release is from water. This was proved during radioisotope. Okay. Where does photosynthesis takes place? The site of photosynthesis is leaf. But when you study the anatomy of leaf, you will come to know that the structure of leaf is complicated. There are, some thylakoids, grana, stroma, which are, having the chlorophyll and they are trapping the sunlight. They are having photosystems also. You would recollect from previous unit that the mesophyll cells in the leaf have a large number of chloroplast. Okay. Chloroplasts align themselves along the walls of the mesophyll cells. Done. And they get the optimum quantity of incident light. You have studied the structure of chloroplast. Studied, huh? Chapter 8. Now, chloroplast is a membranous system consisting of grana. Okay. This is matrix stroma, grana, and that heaps of grana, the collection of grana is thylakoid. Photosynthetic pigments are present over here. Done. The membrane system is responsible for the trapping of light energy, membranous system. Done. In stroma, enzymatic reaction synthesize sugar, which in turn forms starch. Sugar is simple sugar, glucose. Okay. Now the former set of reactions, since they are directly light driven, are called light energy. The latter are not directly dependent on the product. Hence, to distinguish the latter, they are called by convention dark reaction, carbon reaction. Okay. This should not be constructed to mean that they occur in darkness or they are not light dependent. Means within that membranous system, last light dependency is there. But if some reactions are considered a dark reaction, that doesn't mean that these reactions are occurring in dark or they are not light dependent. They are the part of photosynthesis. The photosynthetic pigment chlorophyll have already fetched the adequate sunlight to carry out all the procedures or all the, you know, steps of photosynthesis. Outer membrane, inner membrane, okay, stromal lamella. Now, how many types of pigments are involved in photosynthesis? It is chlorophyll A, chlorophyll B, and xanthophyll and carotenoid. Then now chlorophyll is a very chlorophyll, whether A and B, primary pigment, primary pigment, while other are accessory pigment. Pigments are substances that have an ability to absorb light at specific wavelength as well as to impart color. If there is a pigment in plant, it has two function: color imparting, color imparting. Second is trapping sunlight. Done. Pigments are substances that have ability to absorb light. Okay. Now, the ability of chlorophyll A pigment to absorb light, of course, you are familiar. Wavelength of visible spectrum of light as well as 𝐓𝐚𝐤. Okay. Now, this, understood by your diagram. Now, chlorophyll A, see the wavelength and the light. Okay. Chlorophyll, this is B, this is A. A1, blue light. Carotenoids, yellow light. Done. Now, rate of photosynthesis and absorption. More and more sunlight is absorbed. What will be the rate of photosynthesis? This one. Chlorophyll A, B. These graphs together show that takes place. Most of the photosynthesis takes place in the blue and red regions of spectrum. That is A and B. Okay. Though chlorophyll is the major pigment responsible for trapping light, other thylakoid pigments like chlorophyll B, xanthophyll, carotenoid, which are called accessory pigment, also absorb light. These chlorophyll A and B are primary pigment. They should be present. If they are not present, there is no sense. Who will going to trap the sunlight? If chlorophyll pigments are not present, it is not possible for the green plant to trap the sunlight. Okay. Then other are accessory pigment. What is light reaction? Light reaction or the photochemical phase that in light absorption, water splitting, oxygen release, and the formation of high energy. Done. Pigments are organized into two discrete photochemical light harvesting phases within the photosystem 1 and photosystem 2. These are named in the sequence of their discovery. LHC are made up of 100 of pigment molecules. Each photosystem forming a particular bundle, that is antennae. These pigment help to make photosynthesis to absorb light of various wavelengths. The reaction center is different in both the photosystem. In PS1, the reaction system chlorophyll A has an absorption peak at 700 nm. Hence it is 7p700. Done. The same p680. Electron, what do you mean by electron transport? What do you mean by electron transport? In photosystem 2, the reaction center chlorophyll A absorbs 680 nm wavelength of red light, causing electron to become excited. Jump. These electrons are picked up by an electron acceptor, which passes them to electron transport system. Now, the movement of electrons are always downhill. The movement of electrons are always downhill, downhill, downhill. Okay. Now the electrons are not used up as they pass through the electron transport chain. Okay. But are passed on to the pigments of photosystem 1. Simultaneously, electrons in the reaction center of PS1 cited at 700 ml transfer the electron. These electron move downhill. Molecule of energy NADPH plus required. This whole scheme of transfer starting from PS2 uphill, the acceptor down the electron chain to PS1. PS2 electron transporting PS1. Okay. Now this graph, this movement of electron representing the Z is okay. The shape is formed when all the carrier placed in a sequence on a redox potential. Now, splitting of water is seen in photosynthesis system. Okay. Done. Now, you would then ask, how does PS2 supply electron continue? The electron that were moved from PS2 must be replaced. The splitting of water associated with PS2, water split oxygen electron. This create oxygen, one of the net products of photosynthesis. Net product of photosynthesis is oxygen, should be released to maintain the gaseous cycle. We need to emphasize here that the water splitting complex associated with the PS2, with the PS2, the PS2. Cyclic and non-cyclic photophosphorylation. Done. Living organisms have the capability of extracting energy from oxidizable substances and store this in the bond energy. Special substances like ATP carry this energy in the chemical bond. The process through which ATP is synthesized is named as phosphorylation. Photophosphorylation means synthesis of ATP from ADP and inorganic phosphate in the presence of light. That's why light. Light plus. Presence of light is there. Okay. Now, when the two photosystem work in a series, first PS2 and then the PS1, electron transport occurs from PS2 to PS1. Process called non-cyclic photophosphorylation. The two photosystems are connected through an electron transport chain. Both ATP and NADPH plus H plus are synthesized by a kind of electron flow. When only PS1 is functional, when only PS1 is functional, the electron is circulated within the photosystem. A possible location where this could be happening is in the stroma lamellae. Okay. Excited electron does not pass on to the NADPH. Cyclic flow hence results in the synthesis of ATP, but not to NADPH plus H plus. Okay. Chemiosmotic hypothesis. Chemiosmotic hypothesis. What is chemiosmotic hypothesis? Let us now try and understand how actually ATP is synthesized in the chloroplast. Now, the chemiosmotic hypothesis has been put forward to explain the mechanism. Like in respiration, photosynthesis to ATP synthesis is linked to development of proton gradient. Okay. This time, these are the membranes of thylakoid. Done. There is no difference though they are the proton accumulation toward the inside of membrane. Okay, let us understand what causes the proton gradient across the membrane. Okay, we need to consider again the processes that during activation of electrons. Since splitting of water molecule takes place on the inner side of the membrane, protons or hydrogen ion that are produced by splitting of water accumulate within the thylakoid. This one. Splitting of water is occurring, H plus ion is accumulating. As electron moves through the photosystem, protons are transport across the membrane. Membrane is allowing the transport. This happens because the primary acceptor of electron, which is located towards the outer side of the membrane, transfer electron not to electron carrier but to an H carrier. Inside H2O splitting, H plus ions is forming outside proton. Then outside proton. Okay. When this molecule passes on its electron to electron carrier on the side of the membrane, the proton is released to the inner side or lumen side of the membrane. The NADP reductase enzyme is located. NADP, NADP reductase enzyme is located on the stroma side of the membrane. On the stroma side of the membrane. Then, along with electron that come from acceptor of electron, protons are necessary for reduction of NADP plus to plus H. Now, proton. And hence, within the chloroplast, protons in the stroma decrease in number, while in the lumen, accumulation of proton, because inside lumen everything is creating. Outside proton acceptor is present. This create a proton gradient across the thylakoid membrane. Done. This gradient is important because it is the breakdown of this gradient that lead to the synthesis of ATP. Now, the ATP synthase enzyme consists of two parts. One called the CFW, other portion is CF1. CF1 is thylakoid, embedded in thylakoid membrane, result in a proton gradient. The other portion is called CF1. The breakdown of the gradient provide enough energy to cause a conformational change. Chemiosmosis requires a membrane, proton pump, and a proton gradient. Energy is used to create the pump. Gradient pump is used to create the gradient. ATP synthesis has a channel. Along with the NADPH produced by the movement of electrons taking place, stroma responsible for fixing CO2. Now, fixation of CO2 should occur. H2O should, water splitting should be occur. So if CO2 fix, then only sugar will produce. And if H2O is splitting, if water is splitting, then oxygen. Okay. Where are the ATP and NADPH used? It all used under the phase that is biosynthesis. Okay. This process does not directly depend on the presence of sunlight. Okay. Immediately after light becomes unavailable, the photosynthetic process continues. Now, let us how the ATP and NADPH are used. The use of radioactive 14C/Hem in algal photosynthesis. Okay, this part is not, very much dealing with, but only you have to understood that ATP and NADPH, this process does not directly depend on the this line is only important. This process does not directly depend on the presence of light, but is dependent on the products of the light reaction. Products of light reaction, not light is required. Scientists tried to know whether all plants have PGA as the first product of CO2 fixation. Okay. Now, The first stable product of CO2 fixation was again a then again a sign of production of an organic acid, but when which had 4 carbon atom. This acid was identified to be oxaloacetic acid, OAA. Question comes in. Now, then CO2 assimilation during photosynthesis. Those plants in the first product of CO2 fixation is a C3PGA, C3 pathway called as, then the product formation is C4, so it is 4 pathway. The primary acceptor of CO2 depends, or we can say the dark reaction. The primary acceptor is 5 carbon keto sugar, very important. Ribulose bisphosphate. Now, they also believed that since the first product was a C3 acid, the primary acceptor would be 2 carbon compound. Identify 2 carbon compound before they discover the 5 carbon. Now, what do you mean by Calvin cycle? Calvin and its co-worker, they, understood a cyclic chain. Understood a cyclic chain. Okay. Which actually telling the RUBP regeneration. Okay. RUBP regeneration. Now, Calvin pathway occur in all Calvin pathway occur in all photosynthetic plant. Okay. For ease of understanding, the Calvin cycle can be described. Okay. Under 3. Carboxylation, fixation of CO2, very crucial step. Which enzyme is utilized? RUBP carboxylate. RUBP carboxylate. Okay. Since this enzyme has an oxygenation activity, would be more correct. Enzyme uses carboxylase oxygen. Rubisco. Reduction. These are series of reaction lead to formation. Then these step involve the utilization of 2 molecules of ATP, 2 molecules of NADPH2, the fixation of 6 molecules of CO2 and 6 tons of cycle. 6 molecule of CO2, 6 tons of cycle result in the formation of 1 molecule glucose. Okay, means CO2 1 ton, CO2 second ton, CO2 third ton, 4, 5th, 6. 6 cycles for CO2 results in formation of done. Regeneration. Regeneration of the CO2 acceptor molecule RUBP is crucial for the cycle. Regeneration step require 1 ATP for phosphorylation or to form RUBP. So carboxylation important step, then reduction. Okay. So this is all about this chapter. Today we have completed photosynthesis. Tomorrow we are going to deal with the chapter respiration in plants, the third chapter for plant physiology. Okay students, thank you so much.